Okay, so we have this long title that actually Sonia wrote, but well, but I thought that I could use it for guiding this presentation that basically explains some of our lines of thought and what we do. Okay, and before starting, I'll do this because if not, at the end, it's always I'll forget usually. So here is the place where I work. From the airport, when you come to the city center, you can see this building and the big sign. And here you have some of the students. Well, and it's not in the images, but those are some of my previous students that have contributed to some of the data I'll show you. And of course, I need to make sure that all the logos are in. If not, I'll get punished. Okay, so we're talking about the vestibular system, and you know all about it, but I thought that I could show some of our own images that are always nice. And so you see what a real crista, saccule, and uticle look like from a rat. Those are not human, those are rat epithelia, and the real shape of the vestibular hair cells and the otoconia. Okay. Now, remember that the sensory hair cells are transducers that will allow the entry of, no, this doesn't work. There's an entry of potassium when the stereocilia bend in response to gravity, and then they will release glutamate that will generate the action potentials in the postsynaptic neurons. Okay? We need to remember that we have two types of hair cells, type 1 and type 2 hair cells, that they have very different, or not very different, but somehow different shapes. Type 1 have this amphora-like shape, and they are encased by the afferents that are named calyx afferents because of their shape. And one thing that we need to stress, and I think this is not that common knowledge, is that type 1 hair cells and the postsynaptic calyx are glued one to each other by a special adhesion mechanism that you see at the electron microscopy level as this thick membrane to membrane contact between the hair cell and the postsynaptic neuron. While type 2 hair cells that are more columnar shape, they have standard synaptic buttons contacting them. Okay? These two types of hair cells, I've been teaching sensory physiology for a long time, and I used to say, well, we don't know exactly what they are used for, but or what's the difference. But now we have some new evidence that really support the fact that type 1 hair cells can work faster, do not saturate the system, so are the ones that will encode for the fastest accelerations, and that will support the encoding of the fast movements, while type 2 hair cells will respond more to tonic accelerations. And another knowledge we need is that the three epithelia, the crista, the uticle, and the saccule, have different regions that we can name the center or stiola region in the uticle and saccule, and a periphery. And in the center and the periphery, there are some differences in the distribution of the afferents and on some biochemical properties of the cells. For instance, you see this oncomodulin, which is a calcium binding protein that is present only in the hair cells in the stiola and not in the periphery. And the two types of hair cells, in fact, make synapses to three types of afferents. You have afferents that make contact only with type 1 hair cells, and usually they contact 2, 3, or 4 type 1 hair cells, and those concentrate in the central regions. And then you have afferents that make only button contacts with type 2 hair cells, and those are more peripheral. And then else, everywhere in the sensory epithelia, both in the center and the periphery, you have plenty of dimorphic afferents. Actually, those make 90% of the, I think, or 90, yeah. 70. Yeah. 70% of the dimorphic afferents make both contacts with type 1 and type 2 hair cells. So this makes more complicated to understand how this works. Okay? Now, we have differences. The different types of cells are recognized not only at the ultrastructural level by the shape, but also by specific molecular markers. So we can label them with antibodies against specific proteins, and you can classify them. And we notice that actually the classification that was available by Anna Lisakowski was quite old, using some kind of old immunos, so we could use the confocal with good markers now, and also count all cells in the epithelia. So we did it, and we saw that the composition of the utricle, the saccule, and the crista in terms of type of cells is similar. So we have type 1 hair cells that have osteopontin, or SPP1 is the same, and this calcium channel, PMCA2, in the stereocilia, and they are, the calyx is labeled by this adhesion molecule Caspr1, and this makes about 65% of the hair cells. Okay, and then the type 2, there are two subtypes. Many of them have actually about 85% of the type 2s are labeled by calretinine, and there's a subpopulation that we first named Myo7 only hair cells, but then we saw that they label also for SOX2, that we have about 4 or 5% of type 2s that don't label calretinine, don't express calretinine. So we many times use calretinine as a marker for type 2, knowing that about 15% of them will be missing in our counts. And so we can look and count all of them. Okay. Now, so now tools to prove vestibular function and dysfunction from behavior. Let's start behavior in non-human models. I think this is in this audience, this is already known information. We want to know animal behavior to match human behavior, so we can infer our molecular and cellular data from the animal models to the human. The rationale is well understood in this audience. But one thing is that we want that the human and animal functional measures are equivalent, or that we know how they match. So the inference from the molecular data in the animals is good for the human situation. Okay. Now, so what you do in human? Well, Herman explained very well to you all the things you can do, and there are many other methods, but you can assess vestibular function in deep and in a very precise manner. So there's been a lot of efforts by many people trying to reproduce the same good models in animal models. So if in humans you you study the vestibulo-ocular reflex, so you want to get the vestibular-ocular reflex in rodents, in rats and mice, and well, you can do it. It's doable, but it's quite complex. So actually, few laboratories do it, and they require equipment, knowledge, and also many methods are invasive because the animals need to be fixed. So they put cargols, they put snails on the skull to keep the head still. And now there are options, and this is an image from a paper Vincent is sending that well, this is non-surgery model of holding the rat. Then there are the vestibular evoked potentials. Again, even fewer and fewer laboratories know how to do it, and you need special holders. And in the recent years, there's been publications on the recording of vestibular evoked myogenic potentials, either ocular or cervical, in rats. And first in guinea pigs and rats, and I think also in mice. And this is something quite new in the field that is evolving quickly. Particularly, there has been one paper from the US and 2 or 3 from China doing these recordings in a way that look more consistent than previously. Now, the same idea has been applied to the zebrafish. Berta this morning already showed you this model in which you study the vestibular-ocular reflex in zebrafish larvae, and then at the same time you can do functional analysis of neuronal firing in the brain of those animals. And another approach has been also for approaching the postural changes in vestibular patients, so you can assess locomotion and posture in rats linked to vestibular function. And this is a paper, it's 2016 already. Wow, time goes by fast. So from a French group led by Guillaume Duguet published this paper, and actually they are now trying to combine this system with the assessment of the vestibular-ocular reflex in Paris with Mathieu Berenec, and this is where we plan that Anna will visit to learn about these things. So we'll see. However, a long time ago, when I first met the vestibular system, I didn't know about this, and I didn't have equipment and knowledge. And also I was from the toxicology field, where a main idea was how in preclinical drug evaluation you can quickly determine toxicity, neurotoxicity, or, by the way, vestibular toxicity. And the fact is that vestibular deficient animals are easy to spot if you know what. Oh, the video is not working. Well, can you try? Yeah. So, but you saw this kind of videos, but yeah, you saw this kind of videos of the Walsing syndrome. But actually, doesn't work. Okay, but. You've seen these spinning rats, and now this is common knowledge, but I started recording this in 1993, and this was actually, I had the behavior, and finally we got the hypothesis that this could be vestibular toxicity, and we proved it, and actually we took vestibular toxicity into mainstream toxicology, and so it's easy to spot, but how do you measure it? And we actually, I did a list of ways on how we can measure the tail leaf reflex. This is the contact inhibition. Anna was, well, those are the two tests Anna has explained already. And the initial approach was simply to rate 6 items from spontaneous and reflex behaviors, and each one of them rated 0 to 4, and then this adds to 24, so you could measure vestibular dysfunction 0 to 24. And I would like to point out that one initial thing was the swimming test, and we don't use it because I thought it too stressful and unnecessary. Okay. Now, the other thing we did in these years was to evaluate whether really those rats had vestibular problems, vestibular damage, and we saw that they had. So we used first electron microscopy, either scanning or transmission electron microscopy, and we saw the loss of hair cells in the crista here in this example, and the degeneration of the cells by either necrotic or apoptotic patterns. And so we had this evidence of the damage, and actually we correlated the dose response in terms of behavior and in terms of damage. So we had this link between behavior and damage. That was one of the things. And the question important for me at that moment was that this method of looking at the animals could be used in when evaluating drug toxicity to predict whether this new antibiotic would be ototoxic or not. Okay, so that was one point. Now, this is what we started with Vanessa Martins Lopez on how we could improve those measures, and we came up with this initial, you know already that we use this angle as a quantitative measure of the tail leaf reflex, and we took the time to write as a measure of the eye writing reflex, and the goal was to be fully quantitative and more meaningful. And this was only possible when the cameras to record at 240 frames per second, which all of you have in your mobile phone, became available at affordable prices. Okay, so using these methods, so we could either, for instance, evaluate dose-response relationships and time course analysis of the vestibular loss. And for instance, we describe a situation in which chronic exposure to a toxic compound would induce reversible vestibular dysfunction. And so those were useful methods that we used. Now, as Anna explained to you, she's now using the gravity vector as the axis to which the angles and the times refer to, and is improving within this project. She's improving those methods, and I think that this is going to be a nice contribution to the knowledge of the reflexes and what they mean. Now, the question is that we said we can measure behavior somehow, and we see hair cell loss. How do they relate? So that opens a window to understanding how the vestibular pathology, or the pathology of the vestibular epithelium, translates to functional loss. Okay, so we use this immunohistochemistry, in which we had these populations of defined type 1, type 2 hair cells, and use this knowledge to see, for instance, loss of hair cells, not only at large, big numbers, but also per type and per epithelium and per epithelial region. So one thing that was well known in the field, but actually had been very few times quantified, is that type 1 hair cells in many settings of or many ototoxicity models, type 1 hair cells tend to degenerate first than type 2. And you see here how type 1s, for instance, in the cycle, so those are type 1s, and type 2, if you look at the dose response, you have here effects at 3 to 125. You have no effects on type 2 in the periphery of the cycle. And then you have differences between the utricle and the crista, which are more sensitive, the center. The peripheral regions are less sensitive, and the cycle is more resistant, is less sensitive than the crista and the utricle. So you have these differences in sensitivity on how a toxic compound causes hair cell degeneration. So you can relate this data to the loss of hair cells to the functional data in terms of tail leaf angle or eye writing time. And when you do this and doing some modeling of how the reflexes relate, just correlation analysis and modeling the shape of the regression, you see these kind of graphs coming out that tell you that type 1 hair cells relate much more to the loss of the tail leaf angle and to the change in eye writing time. But you see no relation to type 2 loss. So this strongly suggests that those two reflexes that we are measuring are informing on type 1, not on type 2 function. Okay, and also that the tail leaf reflex relates more to crista or utricle, because this is correlational, so we cannot discriminate between crista and utricle, but we can exclude the saccule and the types 2. And for the eye writing time, it's the utricle and saccule that correlate to the loss of function, not the crista. So you can basically lose most of the cells in the crista, and you still have a writing time that is almost normal. Okay, so that's the kind of knowledge we've been able to generate. And so the question is that we propose that those reflexes are relevant to this question of tripping, and if you trip, do you have a forearm reflex, or you don't have it and you break your head? Okay, so that's different vestibular function. So what this data suggests is that to preserve this falling reflex, you need good type 1 in crista and utricle, and those are precisely the hair cells that degenerate first. So for instance, in the field of regeneration, there are some small success in regenerating type 2 hair cells. And they say, oh, well, we had some additional types 2 hair cells, but the function didn't change. Well, this function didn't change. Maybe other functions did change. So that's what we think we can contribute to the knowledge. Okay, now we move to the more molecular and cellular pathology. So we had the behavior, we had the cells, the relationship between the one and the other. Now we got deeper into the question of what may happen in different situations and even at the molecular level. So I told you that that we have this model in which the control vestibular epithelia can be damaged through hair cell loss that occurs by a quite funny process, which is extrusion from the epithelium. When you have a chronic toxicity that stays longer than the reversal point. But if you intoxicate the animals with this compound for only 4 weeks, then you get a full recovery in those functions. And what we observe at this point is that there's a detachment of the type 1 hair cells from the calyx units and a breakdown of the calyx units. And this is the loss of this adhesion protein, the Caspr1 protein. And actually we were able to see also the pre and postsynaptic sites of glutamine release using this immunostaining, and we saw the decrease in synaptic contacts between type 1 and type 2. So in chronic ototoxicity with the ADPN model, with this particular toxin, we see that the cell under the toxic stress, the type 1s, detach from the calyx and make what we call a synaptic uncoupling. So there's no more signal from the type 1 hair cell to the postsynaptic neuron, and this matches the time when you see the changes in behavior. Okay, so the early effects, the early vestibular effects associate with the detachment. And this is largely reversible. So you can get the adhesion molecules back, you can have repair, you can have complete or partial repair of the synapses. And at the electron microscopy level, you see how the density rebuilds. So the stress hair cell that has detached is able to reattach, and this explains why you get the recovery. Okay, so when you have a patient that is under an ototoxicity treatment and he's having vertigo and he feels sick or whatever, and would I recover? Well, it depends on whether the progression of damage has gone through the point of no return, or whether you are in still this early phase where you can still recover. So you may know that there's little chance for regeneration in the vestibular system, but there's chance for repair if you stop the intoxication soon enough. Okay, so that's something that I think that we learned from all these experiments. Now, well, that was an experimental model that we used for a long time. So we made this apply to the real world. Well, actually, we were able to replicate some of these results in rats exposed to streptomycin. So this is a real human ototoxin. So it seems that we can have also uncoupling of type 1 hair cells from the postsynaptic neurons in people, I would say, exposed to streptomycin, and there's a chance for repair if the hair cells are still in place and not severely damaged. And also, we got access to vestibular epithelia from vestibular schwannoma patients that get the vestibular schwannoma removed by an labyrinthine approach. So they basically make an inner ear ablation. So in this process, the ETNs and the Trias i Pujol Hospital in Badalona will take the vestibular epithelia and send them to our lab, and we do the similar stainings that we do in those rat samples. And we saw that this is Caspiar staining, and it's fragmented. And then we have calyx units that label calretinins, or are calyx-only units that have no Caspiar label. So those are uncoupled units. So we think that this detachment of type 1 hair cells under the detachment of the type 1 hair cells with the postsynaptic neuron may occur also in vestibular epithelia under different kinds of stress, not only toxic stress, but perhaps inflammatory stress because of the presence of a nearby vestibular tumor, vestibular schwannoma. Okay, so this led to this overall hypothesis that we may have characteristic response of the hair cell under stress that may do a few things before it starts being eliminated from the epithelium. And this would be this detachment, but we thought that there could be specific gene expression responses so that we could identify some of the things the hair cell does. Okay, so and also we thought that this was already the moment where we should develop methods to study this in more molecular terms. So this is the first thing we did. We studied several different ototoxicity models using IDPN and streptomycin, rats and mice. We did a whole epithelium RNA sequencing to get the genes upregulated or downregulated in response to the toxic stress, crossed all the models and got a list of 34 genes that are downregulated in common in all models. And I would say all of them, at least the 29 that have been identified, are genes specifically expressed in hair cells, and most of them specifically in type 1 hair cells. So it seems that the hair cell under chronic stress would trigger a program of detachment, change in gene expression that would eliminate specific proteins that that the hair cell needs to be a hair cell. So you have a decreased expression of proteins of the transduction machinery, of the stereocilia, of these things that make the thing a specific, the hair cell a specific hair cell. And we prove that this is associated with real effects at the protein level, not only gene expression that could be linked to hair cell loss. Actually, Anna did some of the hair cell counts to prove that we didn't have significant hair cell loss in this project. But for instance, this PMCA2 protein is a calcium pump that extrudes calcium from the stereocilium to the endolymph to recover during transduction. If this doesn't work, the hair cell can work properly, and actually blockage of this channel may trigger hair cell apoptosis. So I think that we have this lead of how the damage in the hair cell progresses to final functional loss. And there are two sides that we came following this study. First, that we identified new genes. For instance, this new gene is extremely interesting because it's specifically expressed in type 1 hair cells, and it's extremely highly expressed, and it is expressed during development in zebrafish at the very early stage of development, and nobody knows nothing about it. And it seems that it is a gene that is almost not expressed elsewhere in the body. That's a highly specific gene protein that deserve more study. And the other thing is that we develop an in vitro model linked to the vestibular schwannoma study where we thought we could test tumor secretions or other potentially damaging factors. And here, I think this is an extremely interesting result, is that in this vestibular 3D culture, we reproduce well the in vivo situation. We have crista, utricles, and sacules, and we have long-term survival of the cells. We get them at postnatal day 0 in rats, and we are able to culture them for 1 month at least, and they mature in vitro. So we can test then 1 week exposure in mature hair cells, not developing hair cells. And in this model, see that TNF-alpha, an inflammatory factor, kills hair cells when exposed for 7 days, and interleukin-6, another inflammatory cytokine, kills hair cells at extremely low doses, clinically relevant concentrations, I would say, equivalent to plasma concentration. So and these effects match also what you see with the streptomycin. So it seems that both not only ototoxin drugs, but also inflammatory factors can have a deleterious effect on the hair cells. And in fact, the final result that I like a lot is that, not yet, is that this decrease in the expression of VSIG10L2, the new gene that we don't know nothing about, is very well reproduced in inflammatory after exposure to TNF-alpha. So the gene expression response we identified in hair cells using ototoxicity models. In vitro, we can reproduce it using inflammatory factors. So it looks like we have something about the real response of the hair cell to stress, that you can stop and get repair, or you can progress until the hair cell is gone. Okay, so so this illustrates how we measure behavior, hair cells, molecular level, and try to match everything for understanding something about how the vestibular system gets damaged. And actually, one of our goals would be the protection of the hair cells, in aging or in front of stressing factors, environmental factors. Okay, thank you. Questions?